Welcome to the Protein Synthesis series. This video focuses on genetic mutations. At the end of this lecture, students should be able to understand how genetic mutations affect the structure and function of DNA. Mutations can occur in the DNA and then be transcribed into the mRNA, which leads to the wrong polypeptide. Or mutations can occur in the mRNA and be translated into the wrong protein. When looking at a polypeptide or amino acid sequence, the order of amino acids, as dictated by the mRNA sequence, is critical for the function of the protein. Point mutations are a change in the amino acid sequence at only one nucleotide base. Mutations can include substitutions in which one base is replaced with another base. They can also include insertions, in which case one base is inserted between two neighboring bases in the DNA or in the mRNA. They can also include deletions, in which one base is deleted from the DNA strand or the mRNA strand. These changes in the mRNA or DNA can lead to problems with the amino acid sequence. Substitutions create a one-point change in the amino acid sequence. In this example, the anticodons are methionine, which is the start codon, arginine, threonine, and phenylalanine. In the substitution example, the one replaced base changes only one amino acid, causing arginine to become histidine. Insertions and deletions change the reading frame of all amino acids from the point of the change downstream. The slide shows the three amino acids after the start codon are all different from the original mRNA transcript. When looking at the codon UUA, you can see that it codes for leucine. With an insertion or deletion, point mutation, you can see the reading frame will shift downstream. A point mutation in the first base can lead to three possible outcomes. First, a silent mutation, in which the codon still codes for the original amino acid. The protein does not change. Remember, silent, you would not even know it was there. The second outcome would be a missense mutation, in which the base changes, causing the original amino acid, leucine, to change into valine. Remember, this makes no sense because it codes for the wrong protein. The final outcome could lead to a nonsense mutation, in which the new codon is a stop codon instead of the original leucine. Remember that nonsense makes no sense, and it also makes no more protein. Mutations can occur outside of the coding sequence of the gene as well. When there's an error in the promoter, the rate of transcription can be altered. An operator error can also lead to over or under production of mRNA transcripts. Splice site mutations can lead to pre-mRNA being spliced incorrectly, with taking parts of the exons or leaving parts of the introns. Mutations in the translation regulators can alter the amount of protein made. The intergenic regions are between genes, so there's less of a chance that those mutations will alter the expression of a gene. Some spontaneous things can cause gene mutations, such as errors that DNA polymerase did not catch in the sequence complementary pairing, or a metabolic agent that is toxic and either normally converted into another reagent or delivered to the liver for detoxification are instead abundant and can alter DNA. Additionally, isomerization, or the rearrangement of the nucleotides, can cause mispairing. And transposons, or jumping genes, can insert themselves into other parts of the genome, inactivating a gene. There are also induced mutations that can occur, and these are more environmental factors. Things such as chemical factors like cigarettes, hair dyes, commercial cleansers, they can all cause DNA mutation. Physical factors like UV light or X-rays, even disruptions of our circadian rhythm, say from working the third shift. There are biological things like viruses and bacteria that can also cause mutation. Helicobacter pylori is an ulcer-causing bacteria, and HPV, the human papillomavirus, both can cause mutations to our DNA as well. When these mutations occur, it is critical to how they will affect the organism. There are two types of cells in our bodies: somatic cells, which are normal body cells, and germ cells, which are gametes or sex cells, the sperm and egg. So for example, if a mutation occurs in the somatic cells, that would occur post-fertilization, either at some point during embryonic development or during maturation. This mutation would only affect the cells of its descendants. The earlier the event occurs, the more cells could potentially be infected. However, they stay localized to that particular mutant cell area. These types of mutations are not passed on from parents to offspring, so they are acquired. Some cancers are acquired, such as skin cancer, which results from overexposure to UV light. Typically, not one single mutation will cause cancer, but having multiple mutations over a lifetime will increase the likelihood that an individual will develop cancer. Germline mutations in the sperm and egg cells, however, will affect the entire organism and can be passed on from parent to offspring. Because humans are diploid, we have 46 chromosomes, which is 2 sets of 23 chromosomes, one from each parent. When our bodies undergo gametogenesis to create sex cells, since only 23 are put into each gamete cell, there's a 50% chance that the mutation will not be passed on. An example of gametic mutations would be albinism. Some cancer lines are also heritable, which things like retinoblastoma or eye tumors, these mutations can be passed on from parent to offspring, which creates a genetic predisposition in the offspring to getting that type of cancer. Mutations are the raw material for evolution to occur. Not all mutations are deleterious. Some are advantageous and would be selected for in a population, increasing over time. However, other mutations go completely unnoticed in the population, being neither advantageous nor deleterious. Students should be able to contrast point and frame shift mutations. Students should be able to differentiate between missense, nonsense, and silent mutations. Students should be able to describe the causes of spontaneous and induced mutations. Students should be able to indicate possible physical, chemical, and biological mutagens. And students should be able to explain how germline and somatic cell mutations differ.